Hello everyone, welcome back to 8 Exams. This is Maath Alabdali, and today I'm going to go through receptors for A-level biology. And yeah, let's start. Okay, so receptors? So basically, receptors, they are specialized cells. They detect the physical stimuli and they convert them to electrical signal. And each type of stimuli has a specific receptor, and these can be either cells or proteins. Receptors, they use the energy from the stimulus to send the sodium ions into the sensory neuron, creating a voltage change called generator potential, and the generator potential triggers the action potential, and then the action potential is passed through the nervous system. Now, there are many types of response. So there is responses responsible for touch, taste, smell, hearing, vision. In this video, I'm going to go through vision, which is the photoreceptors, the rod cell and the cone cell, and I'm going to go through the touch receptor, which is the Pacinian Corpuscle. And yeah, so if you can see in the top, let me just take my laser pointer. This is the Pacinian Corpuscle, and these are the photoreceptors, which is the rod cell in here and the cone cell. And I'm going to go in depth in this video. So the Pacinian corpuscle is basically a type of touch receptor, okay, and it responds to pressure. Responds to pressure. Yeah, it only responds to mechanical stimuli, so not physical stimuli. So basically, pressure, vibration, and these can be found like in the skin, or it can be found, skin, fingers, toes, these type of areas. And of course it stimulates an action potential. So basically what happens, the corpuscle is compressed by the stimulus. So let's say pressure, you put pressure on your finger, and the lamella, which is, before that, let me just explain the structure of the Pacinian corpuscle. So this is the nerve ending, and these are layers with viscous gel in between, and these layers are called the lamella. Okay, and then this is the blood capillary and the capsule, and this is the sensory neuron, and in between the neurons are of course the nodes of Aurea. And yeah, so an action potential is simulated is when the corpuscle is compressed by the stimulus, and the lamella, it become deformed, changing the shape of the corpuscle. And then after that, stretch mediated sodium ions channel open because the membrane of the sensory nerve cell stretch, and when it stretch, it causes the sodium ion channel. Sorry, this is a mistake. Sodium ion channel to open, and when the sodium ion channel open, the sodium ion diffuses in. Okay, yeah. So the sodium ion move into the sensory neuron, and this causes a generator potential. And of course, the generator potential gets an action potential, as I explained in the last slide. And then the action potential passes along the neuron, and then via the other neuron to the central nervous system, so the brain, the CNS, basically. So photoreceptors, there's cone cell and rod cell. So the retina in the eye, contain photoreceptors that detect light. So the photoreceptors, as I explained, rods and cones, and they contain different light-sensitive optical pigments. So they are sensitive to different wavelength of light. You know how in the electromagnetic spectrum, in the visible light, there's different wavelengths. So they are sensitive to different wavelength of light. And then the light ray basically enters through the pupil and are focused onto the retina at the back in here by the lens, which is this one. And then the photoreceptors in the retina, they produce impulses that travel through the optic nerve to the brain. Okay, that's basically what happened in the photoreceptor. Basically that. Then the light that enters through the pupil is basically converted into electrical impulses, and this is what we call a transducer. So transducer. So when a light impulse is converted to an electrical impulse, is that is transduced. So yeah, and then what happens is that light is absorbed by the light sensitive pigment in the photoreceptors. After that, causes chemical change which increases the permeability of the membrane to the sodium ions, and then the sodium ions rush into the neuron. So this stimulates a generator potential which triggers an action potential and sends the impulse along the bipolar nerve to the brain. So yeah, that's basically what happens in the photoreceptor. Okay, so basically there are two types of photoreceptors as I explained in the last slide. There is the cone cells, the rod cells, and of course they got differences. So in the cone cell, they are basically located in the center of the retina in an area called the foveae. And they give out information in color. Okay, and basically because the reason why they give out information in color is because there are three types of receptor. There is the red sensitive receptor, green sensitive receptor, and the blue sensitive receptor, and these are the three primary colors. So as we know from key stage 3 is that the primary colors can combine to give different colors, and therefore different, yeah, as I said, as I mentioned in here in this point, different intensities of light in these three pigments allow for different colors to be seen. The cone cell, they only detect high intensity. So each cone cell, basically the reason why I'm going to show in a diagram in the next slide is because each cone cell is connected to one neuron. So it takes higher intensity of light to detect. And then the last thing about cone cell is basically it has high visual acuity. So visual acuity is the ability to tell apart 2 points that are close together. So cone cell, cone cell only connect to one other neuron. Okay, this is a mistake. It's not detected, it's connected. So the cone cell is basically only connected to one other neuron. So each individual stimuli can be distinguished. So one cone cell is connected to one neuron, so it can be easily distinguished. It's not like the rod cells, which I'm going to explain. Okay, let me just move to the rod cells and I'll explain. So the rod cell is basically located in the peripherals of the retina in an area called the fovea, and of course it's much more abundant than the cone cell. This is because they're monochronic. So rod cells are monochronic. They give out information in only two colors, black and white, and they can detect both high intensity and low light intensity. This is because many rod cells are connected to one neuron, so many weaker signals can combine to trigger a generator potential. And then it has a low visual acuity, so many rod cells are connected to one neuron, so it's hard to distinguish. So it's basically the opposite of that. So in here, in the visual acuity, in the cone cell, there's one cone cell for one neuron, but in the rod cell, there are many rod cells connected to one neuron. So it's really, really hard to distinguish, if you get what I mean. And yeah. So this diagram summarizes it. So we got the rod cells in here and the cone cell in here. The cone cell, as I explained in the previous slide, it's only connected to one neuron. Okay. However, there are many rod cells that are connected to one neuron. That's why it got low visual acuity, because there are many rod cells connected to it, and then it cannot cannot be distinguished, but it's really hard to differentiate, But the cone cells in here is just one cone cell connected to one neuron. Okay, so and the one more important thing is that you have to know the structure of that. So it's really important to know. So you know how light is light from basically light comes in through the pupil. That's in here, and then over here, it's going to the brain. So an impulse is generated and it's sent to the brain, and this is done through the bipolar neuron, which is linked to a single cone cell. And then over here, the neuron are linked to three different rod cells, as I explained. And then this one is the cell body of the sensory neuron. This is the cell body of the bipolar neuron. This is the synapse, okay, which is the gap in between the gap in between, and then this is the nucleus. And yeah, so thank you very much for listening. See you in the next video, and please subscribe, and also stay tuned because I'm going to upload more of these videos. I'm also going to explain action potential in the next couple of videos, and then this can be linked to action potential. And then yeah, thank you, bye bye.